Welcome to this week's biology lab, which is focused on photosynthesis. Today we're going to be thinking about photosynthesis and how it happens, and specifically designing a simple lab experiment or assay to observe photosynthesis happening in real time. So the way that we're going to do that is by using chloroplasts that have been isolated from spinach leaves. So in this very first part of the lab, I'm going to demonstrate how we're going to isolate the chloroplasts from the spinach leaves here, which are just ordinary spinach that you find at the grocery store. And you'll see this is actually a very simple process to isolate the chloroplasts from these leaves. So the first thing I'm going to do is pour some sucrose solution into the bottom of this blender here. And what the sucrose is going to do is help to protect the chloroplasts as I pulverize the leaf tissue. Then I'm going to add about a handful's worth of spinach leaves into the blender, and I'm going to now pulse the spinach to break apart the leaves, the leaf tissue, and also break apart the cells, which will release the chloroplast into the solution. Okay, so now that I've broken apart the leaf tissue from the spinach leaves, what I have now in the blender is a mixture of all sorts of debris, cellular debris, that was released when I blended the spinach leaves. In the next step, I'm going to just pour this mixture through a couple layers of cheesecloth. And what the cheesecloth will do is remove any kind of large fibers or larger materials from the broken up spinach leaves. And what we're going to be left with at the bottom of this flask here is a solution that's highly enriched for chloroplasts. So there will be other cellular materials in this mixture as well, but it's going to be primarily composed of the chloroplasts, which also give leaves their green color and allow photosynthesis to happen. Okay, so now we've isolated the chloroplasts from the leaf tissue, and at this point I'm going to set aside the cheesecloth and just pour off some of this chloroplast solution into a couple of flasks. Like you can see, it has this really nice green color because it's completely full of chloroplasts, which are the individual organelles from within the plant cells. So here again you can see the chloroplast solution. And what I'm going to do now, before I keep going, is wrap these in tin foil so that the chloroplasts are protected from light as we set up the experiment. And I'm also going to put them on ice so that they will retain their integrity of the organelle as we set up the experiment. So now I've got these two flasks with the chloroplast solution in them, and the first thing I'm going to do is add detergent into one of these flasks. I'm going to add about 5 milliliters of detergent. And this is going to be to test whether or not the detergent is going to interfere with the function of the chloroplast. So I'll explain that more as we continue to set up the experiment. So at this point, what I'm going to do is set up 4 glass test tubes that are going to be the basis for our experiment. So there will be 4 little glass test tubes here that I've labeled 1 through 4, and each of these test tubes is going to have 3 different main ingredients added into it. There's going to be, first of all, about 2 milliliters of water that just provides the base for this assay. There's also going to be 0.5 milliliters of tris buffer. This buffer will stabilize the pH of our assay and keep the chloroplasts at a physiological pH where they should be active. And then we'll also be adding some of this chemical here, which is called DPIP, which is a color indicator. So this DPIP is really the key chemical that's added into these test tubes that's going to allow us to see whether or not photosynthesis is occurring. The way this will work is, once I add the DPIP to each one of the tubes, each one of these 4 tubes is going to start off blue because this DPIP itself is a blue chemical. But what happens is, if photosynthesis occurs when I add the chloroplast into these tubes, the DPIP will start to lose its color and it will fade and maybe even become completely colorless. So what this is a chemical color indicator that we're going to add into our reaction tubes here, and if the color stays blue, that means no photosynthesis has occurred, and if we see the color changing and fading to become clear or having no color, then we know that photosynthesis happened. And you can read more about how this reaction works, but the basics of this reaction are that as the chloroplasts photosynthesize, they produce byproducts and they release electrons that interact with the DPIP. When the DPIP is in its blue state, it's oxidized, and that's when it appears blue. And if the DPIP becomes reduced as a byproduct of photosynthesis happening within the tube, then it's going to lose its color in that reduced state. So that's the basics of how this assay is going to work based on this color indicator. And now I'll take you for a close-up view of these 4 tubes and show you the next couple of steps. Okay, so here's a close-up view of our 4 glass test tubes that are going to be the reaction tubes where we're going to run our assay. Again, just to reiterate, each one of these tubes has a mixture of water, buffer, and then the DPIP chemical, which is the blue chemical that will turn colorless when photosynthesis is occurring. So now into each one of these tubes that I've labeled 1 through 4, we will be adding different combinations of chloroplasts or applying different experimental conditions to test and see what's happening with the chloroplasts. So tube number 1, I'm actually not going to add anything more to that tube. In this experiment, tube number 1 is acting like a negative control here, this tube number 1, because I'm not going to add any chloroplasts and I'm not going to do anything else to that tube, so it's going to act as a baseline for comparing the colors of the other tubes. Into tube number 2, I'm going to add a drop or 2 of the untreated chloroplasts. So that will be from one of these flasks that I did not add detergent to, of the chloroplast mixture that I just isolated. And I'll do that using an eyedropper. So that tube will also be allowed to be exposed to the light. So that's a tube where we might expect to see a color change because we have untreated chloroplasts that should be capable of photosynthesis, and we're also going to allow it to be exposed to light. So photosynthesis should occur and we should see a color change or fading of the blue color here in tube number 2. Tube number 3, I'm going to choose in this experiment to add a drop or 2 of the treated chloroplasts into tube number 3. And I'm also going to leave this tube in the light. Those treated chloroplasts are the ones that I added detergent to, which we might develop some hypothesis as far as what the detergent is going to do to the chloroplasts. Perhaps it's going to cause the chloroplast to lyse or otherwise inhibit the activity of the chloroplasts. And if that's the case, then this tube should either stay blue or should show less of a color change than we're going to see in tube number 2 that contains the untreated chloroplasts. And then finally, in tube number 4 here, we're also going to add chloroplasts, one drop, a drop or two of the untreated chloroplasts, but in tube number 4, I'm actually going to block the light. So I'm going to take some tin foil here and just wrap it around tube number 4. And what I'm trying to do is prevent any kind of light from getting into the tube. So I've wrapped the tube, and now I'm going to make also a little cap for the tube that's also made out of tin foil, something like this, so that we can really try to block the light from entering tube number 4. Okay, so we've got our 4 tubes, and I'm ready to start adding some chloroplasts to the different tubes. And I have the tubes up against the white background so that we'll be able to watch this assay unfold here right before our eyes. So I'm using a simple eyedropper to dispense the chloroplast solution into the tubes. Before I do that, I'll give you another good look here at the color and the clarity of the chloroplast solution. And I'll also put up an image on the video of what these chloroplasts from the spinach leaf look like under the microscope. They're basically little small discs, these tiny organelles from within the plant cells. So again, I'm going to start off by putting 1 or 2 drops. This is the untreated chloroplast into tube number 2. So just like this. And I'm also going to, while I'm at it, open up the cap for tube number 4 and put in the same 2 drops of chloroplast into tube number 4 and then close it up right away so that light doesn't get in here. So that was the addition of the 2 untreated chloroplast solutions. Now what I'm going to do is clean out the eyedropper and add another 2 drops of the treated chloroplast into tube number 3. Okay, so here comes the 2 drops of treated chloroplast into tube number 3. These were treated with detergent. And we're going to watch this start to unfold here, and I think what I'll do to sort of speed things up is shake all of the tubes a couple times just by inverting them, and I'm also going to do that for the control tube just so that all the tubes are being treated the same. Okay, so I added a light here, and the reaction has been going for a couple of minutes now, so that you can hopefully see the differences in color between the 4 tubes, or right now the 3 tubes. You can see that number 1, which is again our negative control, is still blue. Number 2, you can certainly see a visible color change. It's starting to turn sort of teal, and if we allowed this experiment to continue, that one might eventually turn completely clear. Number 3, with the treated detergent, we don't see a color change there. It looks pretty much just like number 1, the negative control. So for that one, we would expect that the detergent did inhibit the activity of the chloroplasts, since it looks like just like the baseline. And now I'm going to also go ahead and remove the tube from the tin foil, and we'll see what happened with the tinfoil tube. So here's the experiment that ran without light but did contain the active chloroplasts. So it's the same as number 2, other than the fact that it has been in the dark for the past couple of minutes. And hopefully you can see that in tube number 4 we still have the same overall blue color as you can also see in tubes 1, 3, and 4. So really the only tube where we saw the noticeable color change becoming less blue and eventually turning completely clear if we allowed this reaction to occur for long enough, is tube number 2 with the active chloroplasts. Tube number 3 with the detergent-treated chloroplast has not changed color, and tube number 4, which was protected from light, we also don't see much of a color change. Although now, because it's exposed to light, it's starting to change color fairly rapidly. So this is the basic setup for this type of assay, using this chemical DPIP to assess the activity of chloroplasts and the rate of photosynthesis occurring in real time.